this is question 4, basic agricultural duty. 4.1. A purebred black cow is crossed with a purebred red bull. So here we can see where we have a purebred. A purebred just means homozygous. Here we have a homozygous black cow that is crossed with a homozygous red bull. The offspring of the F1 first female generation have black color. That means the black color was homozygous dominant, and the red color was homozygous recessive, because the allele for the black color, okay, was more dominant. That means the black color was able to mark the recessive allele for red color. Okay, was able to mask the red color. Okay, so we ended up having the offspring that all have the black color. That means black was dominant over the red color. Use the letter B, okay, and yeah, use the letter B, which is dominant B, and this is the B to answer the question. 4.1.1 indicate whether black or red color is dominant. Okay, this one days you can see whether the offspring are what? Are all black. That means the black was dominant over the red color. That means the black is dominant. 4.1.2. Why are you saying black is dominant? Do you want the answer? So the allele for black was able to mask the allele for red color. Okay, the allele for black color was able to mask the allele for red color. As a result, all offspring were black because of the black color was dominant. 4.1.3. Determine the following. We want the genotype of the cow and the genotype of the offspring. The genotype of the cow, okay, so the genotype of the cow. The cow is black, no? Black cow, okay? So that means the genotype of the cow, that means the cow has to be homozygous dominant. Why am I saying the cow has to be homozygous dominant? It has to be homozygous dominant so that it will be able to mask the recessive allele of the red color. Let's do a short sketch on the side so we can see what we're talking about. Homozygous genotype. We have black, homozygous, black cow is crossed with a red bull. Okay, the genotype. This genotype we got it because we know the purebred, that means homozygous. Homozygous, that means the two alleles are identical. Okay, two alleles are identical. That's homozygous. Homozygous means the two alleles are identical. Okay, so and here we know black was able to mask the recessive allele. That means the black color was dominant. Okay? Dominant allele. So we have a dominant allele and purebred. It requires both alleles to be identical. That means both alleles must have a capital letter. That means we won't have two B's with capital letter. Okay, that means for cow we provide capital letter B. It's crossed with what? A red bull. A pure breed means homozygote. Two alleles are identical. That means this red bull is masked by the black color. That means this bull is recessive. So if it has a recessive allele, that means both alleles for the red color must be recessive. That means we don't have small letters of B. Then this is our genotype, right? Homozygous, dominant, and homozygous recessive. So it's going to go through meiosis. Okay. Then we're going to have our gamete, then fertilization. Okay, so this one is going to be constant. This one is getting a heterozygous condition for BB, and C is going to be constant. We have a homozygous condition for BB also, and it made our offspring to be identical. Okay, the genotype is heterozygous BB. Okay. The L form genotype, the genotype. Okay, L, B, D. And the phenotype. Okay, let's not try the phenotype. Phenotype, but it is all black. Let's go back to our question. The genotype of the cow. Okay, we look at our sketch here. The question that we made. The cow is here. The cow is having a homozygous dominant allele. Okay, as BB, two capital letter BB, and B says the genotype of the offspring. The offspring is this one. The genotype that we got was in a heterozygous condition, which is capital letter B and a small letter B allele, dominant and a recessive allele. Okay. Yeah. Then 4.2, the diagram below represents a type of crossing between two peer varieties showing two genes. So we have two characteristics, a color and a texture. The color we have green and yellow. Texture we have rough and smooth. Then here 4.2.1 they say they want the type of crossing above. Okay, so we know this is a crossing involving two characteristics. Okay, and the crossing that involves two characteristics is what? Dihybrid crossing. Is dihybrid crossing. So then if it was a monohybrid crossing, it will involve only one characteristic or only one trait. For example, to involve only one color. Right. So what is a trait? Nice term. A trait is a characteristic. Okay. Right. For 4.2.1, we said the answer is dihybrid cross. So 4.2.2 it says, why are you saying the dihybrid cross? You got the answer. Right. We say it is a dihybrid cross because two different characteristics are involved in the crossing. We have the color trait and the texture trait. They are involved. Two characteristics are involved in the crossing. Then 4.2.3 determine two possible phenotypes of the first female generation. Two possible phenotypes. So if you look at this diagram, okay, if you look at this diagram, you should be, I would say, you don't need to be smart to get it right. But if you look at it, you'll see that, okay, this is a combination of characters. And sometimes it can be scary, I know. Yes, it is scary, but we can break it down. Okay, here we have the genotypes of the offspring, but here we just have the genotypes, the genetic composition, the alleles, right? So just have to look at these genotypes and see what the phenotype is by looking at the genotype. Okay, right. So homozygous G and homozygous R dominant. What is this homozygous D dominant and homozygous R dominant? Since here we have the dominant, homozygous dominant for both characteristics, color is dominant and the text is dominant. So we're going to look at G, okay, which is green color, okay. And the second characteristic, we're going to look at R, which is rough text, okay. So we're going to combine this color trait and the text trait. So we're going to say the phenotype is green and rough text. That's why we wrote green and rough. Okay, that's homozygous green and rough text. Then homozygous dominant GG and a recessive RR, and homozygous recessive RR. Okay, we're going to look at dominant G, which is green, and recessive R, which is smooth. Okay, so this this makes green and smooth. Okay, that's why we wrote green and smooth. Okay, makes green and smooth. So this is the physical appearance that we're looking for. Green and smooth. Okay, green and rough. So, but there are many phenotypes here. We can have yellow and smooth. Okay, yes. So they were not constrained, so you were not limited. So any possible phenotype that we can see, it is correct here. And 4.2.4, calculate the percentage of offspring that have yellow and smooth PSCs. Okay, so if we know about the hybrid courses, so not all the hybrid courses, but if we can look, then we see it is that the hybrid course that has 9:3:3:1. That means you already know the answer, because that one is the one that is homozygous recessive for both characteristics. Homozygous recessive for yellow and homozygous recessive for smooth. That means it will be yellow and smooth text. Right. So right. So step one for getting to the answer, we need to understand that a yellow seed is made up of a recessive allele, small letter G, okay? And a smooth is made up of a and a smooth text is made up of a small letter R. Together, this GG and RR are our genotype. Okay, the genotypic makeup, the genetic makeup that will help us to determine the phenotype, which is yellow and smooth C. Step 2. We see that there is only one offspring with yellow and smooth seed. Then we count the total number of our offspring. So if you don't count if you don't count the number of offspring that are yellow and smooth, okay, you can use your knowledge of 9:3:1, and know which is only one, unless they switch up the crossing, I don't know, but most of the time they use this format 9:3:1. Okay, then you know, okay, there is only one yellow and smooth offspring, okay, in this thing, right? Or progeny, only one progeny. So calculate the percentage of offspring that has yellow and smooth PSC. So then after finding out that okay, there's only one offspring that is smooth and yellow, then now we just have to get the total number of offspring. How many offsprings are here? We have 4, 8, 8 plus 8 is 16. Okay, we have 16 and total. So step number 3, we're going to take the number of yellow and smooth offspring, which is 1, then we're going to divide it by the total number of offspring, and we times it by 100. Okay, why are we timesing it by 100? This is because we want a percentage. Okay, so this gives us an answer of 6.4.3. The differences in the phenotypes of plants and animals of the same species are influenced by both the genes and the environment. 4.3.1. Give a genetic term for the phenomenon in the statement above. So let's move back to the scenario. The differences in the phenotypes of plants and animals. The differences, there is variation in these plants and animals of the same species. Variation, okay? Variation. So the correct answer is variation. The difference is variation. But maybe we have our answers, so we need to go a long way. So 4.3.2 name 2 environmental factors that can have an effect on this phenomenon. So variation, what can have an effect on variation? Temperature. Okay, temperature can have an effect on variation. For example, the people that live close to the equator are black, or maybe they have dark black color. Okay, then let's look at the people who live away from the equator, let's say maybe Cape Town, close to the sea. They have this light color, okay, light skinned, right? So so you can see that the temperature can have an effect on variation. Thus you can have those with light skin color, those ones that have brown or black color, then those ones that have the black color, because of the sun. Okay, so A is temperature, or you can see the climate. Temperature and climate is the same thing, temperature of the climate. So B, another thing that can influence this is the feed. or the nutrition, the feeding. So yeah, the feeding can also influence, that can have an effect on variation. So 4.3.3 give two types of selection. So selection, when it comes to selection, we have two types. Right? We have two types of selection. So we have natural selection and artificial selection. Natural selection does where we introduce the survival of the fittest. Okay, the organisms that are best situated to the environment will be able to survive and produce the offspring and pass those traits that made them to survive to the offspring. Okay, so yeah, so the progeny then offspring. Okay, just a reminder, progeny then offspring. Then artificial selection, artificial selection, that's when there is human interference, and these people, okay, they try and find animals or plants that have good desired traits, okay. Then the plants or animals that have good traits will be used for breeding purposes. Animals with good traits will be used for breeding purposes. That is artificial selection. Okay, but here the beginning one need to explain what is natural selection or artificial selection now. Invest into this term so that we can be on the same page. So up to so far, yeah, we are good. Then 4.4, the picture below shows two different breeds of animals, from animals. As you can see, have a dairy breed and a beef breed. 4.4.1 means the breeding system if the animals in the pictures above are allowed to breed. So it's okay. So okay, you have two different breeds, okay, and these two different breeds are of the same species. Okay, if there are two different breeds of the same species, what do we have? Okay, what breeding system can we use? Okay, cross breeding. Okay, cross breeding. Then so yes, they'll ask you to give a reason for our answer. Why are we saying cross breeding? Two different breeds, of which are of the same species are allowed to breed. Two different breeds of which are of the same species are allowed to breed. Okay, because we have a beef breed and dairy breed. So 4.4.4, 4.4.3 really the two advantages of breeding system in question 4.4.1. Two advantages. What is two advantages of this kind of a breeding system? The advantage is that it increases genetic variation in the head. Okay, it increases genetic variation in the head. Meaning the animals will be different. Okay, will be different. The head will have different kinds of brains. Okay, different kinds of brains, which is amazing. Which is point number 2. It produces hybrid vigor. Okay, hybrid vigor basically is when an offspring has an increased performance. Okay, and then offspring with increased performance. Okay, so this is when the progeny is better than the parent. Okay, but that's when the progeny performs better than the parent. 4.4.4 then two disadvantages of inbreeding. Inbreeding basically is when cross animals that are closely related. For example, a daughter and a father, a sister and a brother, okay, half sister and a brother, okay, as closely related animals, okay. Right, so the disadvantages of this inbreeding. So with inbreeding, lethal genes can be introduced, which is okay. Lethal genes can be introduced, and another one is that it leads to inbreeding depression. It leads to inbreeding depression. So inbreeding depression basically is when there is decreased performance of the progeny. Now that means the progeny is performing worse. So or even the parents are better, the performance of the offspring is decreased compared to the performance of the parent. Okay, the performance decreased, the decreased performance of the offspring. 4.5, a homozygous brown E, which is written by the capital letter or the dominant A, was mated with a homozygous white with a recessive A or small letter A, but we prefer using the terms of genetic, which is recessive A. The first filial offspring are all brown. The A letter was dominant over the recessive letter. The dominant allele was able to mask the recessive allele. Okay. All offsprings were brown. The first filial generation of these were mated with an unknown friend, and all the second filial generation of spring were brown, and all of the second filial generation of springs were all brown. Okay, so this tells us that the first filial generation of offspring was brown. Then they took those offsprings, they made them the parents, then they mated them, and also the second filial generation produced brown offspring, all brown. That means we're in this dominancy. Okay, let's go to the question 6. 4.5.1. Draw a pie spot to illustrate the second filial generation. So I don't want the first one, they want the second one. Okay, so basically, if you know the course, you know, okay, if I have a homozygous dominant and a homozygous recessive group, it's called homozygous and pure breed are the same terms. Okay, so homozygous just means we have two identical alleles. If we have dominant allele, the two A's must both be the capital A. If we have a homozygous recessive, the two A's must all be small a. The two of them, two alleles, okay, must all be the same, identical. Okay, so if we crossed homozygous dominant and homozygous recessive, it means we crossed the capital letter E with the capital L. We cross the capital letter E with the small letter E, and as a result we got the heterozygous condition. Okay, if you don't understand how we got a heterozygous condition, that means we are going to make the crossing for the first filial generation on a separate page, on a blank page or screen assignment. Okay, because it's not part of your account. Okay, because say they want the second filial generation. Okay, so let's do it. Right. run the script. Right. Then it should look something like this. Right. So we have dominant A, which is the commits on the top, and recessive values on the vertical side, on the left vertical side. As we know, the E is the female, no? It's the female here, because that's the sheep's, right? The E is the female, right? So the E is at the top, the commits for E is at the top. You have dominant A and the dominant A. So first column dominant A and second column dominant A. Okay, dominant just means that's capital letter. Then the realm is here on the left hand side. Okay. Which can do so over small letter A, small letter H is values. Okay. So to represent name, we use this kind of a symbol here, as you can see. We use this kind of a symbol, and the female we use this symbol. It's like a cross with a 0 on top. Then for male, there is a 0 and this arrow that goes outwards. Right, so we're going to cross the gametes for the female E, which is dominant, with the gametes for the male realm, okay, which is recessive allele. Okay, so it gives us dominant and the recessive A, dominant A and the recessive A, and all of them are going to be dominant A and recessive A. Right. Heterozygous condition. All of them are in heterozygous condition. 1, 2, 3, 4. All of them. So this is the first crossing, and it was not the one that they match here. Okay. Now that we have the genotype from the first crossing, then we go back to read our scenario. The F1 offspring were all brown. That's what we got. Right? Yes. And the F1 is, okay, the F1 is the infant, the female animal. Okay, female animal were mated with an unknown rare. Oh, interesting question. Unknown rare, that means you don't know the genotype of this rare. Okay, and all the F2 generation offspring were brown. Okay, then this has to be a tricky question. Then if there's a tricky question that needs to be system straight. So because of this, we're not going to write the phenotype and the genotype. Let's going to get right into it. Okay. Because the principle is the one that they're going to mark. Okay. Let's draw it and see. Okay. So we have the male and the female. Show these samples and the principle. Alright. So let's start with what we have here. We have a heterozygous allele. Okay. And we know that these heterozygous alleles are going to go through that analysis to give us the gametes. So first column we're going to write capital letter A is dominant A, and the second column we're going to write recessive A. Right? Then we are start to look now. So what will the vertical side, okay, this vertical side on that on our left hand side, vertical side, what should this ball be, so that when we take the gamete from the female and gamete for the ball, it gives us a dominance. Yeah, it gives us dominance. It means the first one, it has to be dominant. Okay, why it has to be dominant? Because we want it to mask this recessive allele for the female. Okay, because this cow has a heterozygous condition. Okay, dominant and recessive. So this recessive has to be masked. Because if it is not masked, then it is tough. So taking the dominant allele from the male parent and dominant allele from the female parent, then we have okay, then we have right. Yes, we have AA, which is dominant. Okay, homozygous right. Then we're going to take a recessive allele from the parent, okay, and a dominant allele from the male parent. This is a heterozygous condition. Alright. Now that means we want the gamete for the second row. Second row, we should have a dominant allele because we want to mask the recessive allele in room number 3 and room number 4. Room number 3 and room number 4. So if the male parent gives us dominant allele, okay, and the female parent gives us a dominant allele, then we have a dominant, which is homozygous dominant allele, and we take another dominant from the male parent, the gamete that has a dominant allele from the male parent. Then we are trying to mask the recessive allele that we're going to get from the female parent. That is a heterozygous condition. So we have two homozygous dominant. Two of our offspring have two capital letters, and two of our offspring have a heterozygous condition, dominant and recessive from this crossing. Now, this unknown ram in the male has to have a homozygous dominant genotype, so that it can produce the gametes of dominancy, dominant A and dominant A in the transfer, so that they can mask the recessive allele for the mother, for the female cow. Okay, and it's not a coincidence. So as a result, this heterozygous and dominant homozygous dominant, they give us what all because this dominant allele and not the recessive allele. Now they want the genotypic ratio of the F2 generation. Okay, so the genotypic ratio we're just going to count. Okay, so just the right A genotypic ratio, then how many homozygous dominant alleles do we have? Homozygous dominant, we're talking about those letters that have capital letters, 2 of them. Okay? We write 2AA. Right. These two heterozygous. So you can see 2 offsprings are heterozygous, one dominant and okay. Then the ratio is going to be 2:2. Okay, is to 0. 2:2 is to 0. 2:2 is to 0. Right. Then the number of white offspring in the F2 generation. So we can have the number of offsprings in the F2 generation because they told us, or make sure in your part score you make it a point that the offsprings are all brown. Okay, and we sure all offsprings are brown, and making sure that they are no ram will produce the gametes that are dominant, so that we can carry the recessive allele. So, and also we saw, okay, there's no recessive allele. The recessive allele has to be homozygous. Okay, have to have 2 small a's. Okay, 2 small a's, so that we can have white card. And now we don't have a homozygous. Don't have a homozygous recessive allele. So the number of white offspring, don't have white offsprings here. We only have brown. All the phenotypes, they make a phenotype of brown, physical appearance of brown. So we don't have white, so maybe this is 0. You can't have white progeny because the dominant allele will mask the recessive allele one cross. Okay, one cross. Then 4.6. Let's talk about the length of a maize plant that is controlled by 3 pairs of genes. Then we will assume that the base length of the recessive, which is small letter A, B, D, D, maize plant is 40 centimeters. Each additive allele contributes 5 centimeters. But I believe this is our base length. This recessive A, B, D. This is the genotype. Okay? 40 centimeters. But this genotype, small letter A, B, D, is equals to 40 centimeters. Then they say if we add the dominant allele, that dominant allele will add an extra 5 centimeters. Okay, right, to what we have, and now we have 50 centimeters. 4.6.1 determine the genotype of the highest maize plant. So before we get into that, we need to know what is this. Okay, this is polygenic inheritance. Okay, where this maize plant is controlled by many genes. It is mostly used in the production of milk, production of eggs, yeah. So each dominant allele that is added will increase the yield of milk. If we were producing let's say we're producing 100 liters per day, then we can add more dominant genes. We can end up producing 150 liters. Let's move on. 4.6.1 determine the genotype of the highest maize plant. So here we are dealing with the maize plant. So the genotype, as I said earlier, I said that this A, B, D, they represent the genotype. Okay, so what will the genotype look like for the highest maize plant? That means we have to substitute this small a's with the future letter A, B, D. Okay. So that means we're going to have dominant A, dominant B, dominant D. That is the genotype for the highest maize plant. Right. So 4 and 6.2 calculate the highest height of the maize plant. Show all your calculations. Also, as of written here, A, B is 40 centimeters. Okay. So this is the base length. The excessive value of this 40 centimeters. Each terminal value as 5 centimeters. So for the highest maize plant, which is this notable the range and 4.6.1. Okay, so to have this genotype, we have to say 40 centimeters onto the 40 centimeters to add 2 dominant alleles for A is going to give us plus 5, 2 dominant alleles for B is going to give us plus 5, and 2 dominant alleles for D, then it's going to give us 5 plus 5. Then we have 40 centimeters plus 5 plus 5 plus 5 plus 5 plus 5 plus 5. So it's equals to 40 centimeters plus 80, and it's equals to 8 centimeters. Right. 4.6.3 name 2 genotypes that will give rise to a maize plant with a height of 55 centimeters. 2 genotypes that will give rise to a maize plant with a height of 55 centimeters. So that means we have to do that now. Okay? So if I have 40, if I have 40, how many 5? How many 5 centimeters do I need to get to 55? I need 50. Okay, so 50 divided by 3 is what is 5. Okay, it's 5. That means I will need 3 5s. Okay, so we are going to look at what represents 5. What represents 5 is a dominant allele. Dominant allele adds 5 centimeters. Dominant A, dominant B, then excessive B, excessive D, excessive D. And this is the method we want from 2. Okay, or we can have capital letter A, capital letter A, small letter B, capital letter D, small letter B. But as long as we have 3 dominant alleles in the genotype, then we should get it right. 4.7 give 3 techniques to genetically modify farm animals. For the farm animals. So these techniques are those ones that are specifically made to modify farm animals, and we have those ones that can modify plants. So sometimes they can make you correct. It depends on the marking guidelines, but for just in case, the electroporation and the gene guide, and the bacterium tumefiscus bacterium tumescentius, gene gum, electroporation. Those ones are for the plants. Photosynthesis is specified for the plant. Okay. Gene gum, electroporation, and the bacterium tumescentius are for plants. So for animals, here I said for the animals, retroviral cells, yes, retroviral cells is the correct answer here. Embryonic stem cells is the correct answer, and also microinjection. Microinjection works for plants and can also be used for animals. Okay, but if you wrote electroporation and gene therapy, they can give you a mark. It depends, but they will not be correct because I can see that here they also listed it in the memo. Okay, so maybe I don't know, maybe they will like it, I don't know, but yeah, you need to be careful, but just know if you are writing the wrong ones, it might be risky, but they can make it correct. It depends on the markers, what decisions are they taking. If most of the learners wrote the correct answer, that means we assume, okay, yes, learners know this thing, then they can penalize those ones that didn't write the right answer. So it depends, yeah, it depends on the marking criteria and what was the decision about the marking guidelines. Okay, so but just to be safe, yeah, this is what we have. We have microinjection and embryonic stem cells, retroviral cells. Yeah, all the best. That's it for now.